In the last lecture, we looked at the rheology fundamentals. We looked at what is rheology and what are different models that can be used to explain the non-Newtonian behavior of complex fluids. We also saw that when there are particles suspended in a simple fluid, the suspension of particles in a simple fluid behaves as a complex fluid. So, as we know that blood is a suspension of different flexible particles in a simple fluid, we need to understand first the different components and their mechanical properties in the blood or their mechanical properties. So, in this lecture we are going to look at the different components of blood and their properties. So, as you can see in this image, if we separate the different parts of blood, it has a clear fluid called plasma, and in this plasma there are three different types of particles called white blood cells, red blood cells and platelets, which are suspended in the blood. They are also known as red blood cells are known as erythrocytes, white blood cells are known as leukocytes, and platelets, they are helpful in the process of, or they are one of the major factor in the process of thrombosis or clot formation. So, they are also known as thrombocytes. You can see from here that plasma constitute about 55% of blood, and red blood cells, they constitute about 40 to 45% of the blood, and white blood cells and platelets, they constitute about 1% of the blood volume. So, let us look at these components one by one. Plasma is an incompressible Newtonian liquid which has a viscosity of about 1.1 to 1.6 centipoise. The viscosity of blood is dependent on a number of factors, on the age of the human, the weather, the geographical conditions, the race, the pathophysiological condition. It can also vary with the day and time of the day. So, it is a very variable property. So, it is not possible to have or give one value of viscosity and there is one range. Nominally, for calculation purposes, it is generally taken as 1.2 centipoise at 37 degrees centigrade, which is body temperature. It is not known what is the how the viscosity of blood behaves with temperature. So, it is generally studied, all the rheological measurements are generally performed at body temperature, which is about 37 degrees centigrade. Okay. The density of plasma is about 1030 kg per meter cube, which is same as that of water, and plasma, apart from water, it contains different kind of molecules, protein, plasma proteins, different blood clotting factor, from blood clotting factor 1, 2, 3, 4 and so on, and sugars, lipids, vitamins, hormones, enzymes, antibodies. Okay. So, among the proteins, plasma contains three major type of proteins. Albumin, which is a small molecule and it provides the colloidal osmotic pressure in the plasma. The another protein which is present in plasma is globulin, and it is a relatively symmetric molecule and it is mainly involved in the transport of lipids as well as in the antibody reactions. The third protein is known as fibrinogen, It is a large asymmetric molecule and it plays a major role in the coagulation of blood or thrombosis process. All of these also affect the viscosity of plasma to an extent. The second major component of blood is red blood cells, and they constitute more than 99% of particulate matter. Particulate matter means particles. So, about 99% of the particles which are present in the blood are red blood cells or erythrocytes. The volume fraction of red blood cells in the blood is about 40 to 45%, and because it is an important factor pathologically, so this has been given a specific name. The volume fraction of red blood cell in the blood is known as hematocrit, which is percentage volume fraction of red blood cells in the blood. The HCT or hematocrit in man is about 40 and about 38 in women on an average. The number of red blood cells per millimeter cube is about 5.2 million in man and about 4.6 or 4.7 per millimeter, 4.7 million per millimeter cube in women. Again, these numbers are only indicative. So, because the red blood cell constitute the major suspended particles in plasma, so they determine, or they are the main factor determining the rheological behavior of the blood. The density of red blood cells is not very different from plasma. It is about 1080 kg per meter cube, whereas that of plasma is about 1030 kg per meter cube. So, on this image, what you see here in part A is a SEM image of RBCs, and you might notice there that the shape of an RBC is a disc shape, which is biconcave, concave on both sides, and the diameter of this disc is about 7 to 8 micron, and 2 micron is the thickness of the disc. So, the red blood cells are disc shaped particles, they are not spherical in shape, and this shape is at rest. So, they can deform from this shape under the flow conditions. As we know that red blood cells, they have hemoglobin in it, and the function of hemoglobin is to carry oxygen to different parts of the blood. So, it should have a very good rate of mass transfer, and mass transfer is dependent on the surface area. The higher the surface area, the higher the rate of mass transfer. So, red blood cell should have higher surface area per unit volume. It is desired. The volume of RBC is about 90 micron cube, and the cell surface area is about 140 micron square. So, the minimum surface area for a given volume is that of a sphere. Let us compare this number with that of a sphere. So, for a sphere, the volume is 4 by 3 pi r cube, and r will be 3v by 4 pi to the power 1 by 3. So, we substitute it here, we will get the radius to be about 2.78 microns. Now, if we calculate the surface area of a sphere of this volume, that is equal to 4 pi r square, we will get about 97 micron square. So, the surface area of discoid shape is about 1.5 times of a sphere of the same volume. And because of this property, RBCs have the ability to change their shapes without actually changing the surface area or the volume. So, they can take infinite number of shapes, different shapes, without having extra stress on the membrane surface. Okay. RBCs, the membrane of the RBC, it is a viscoelastic solid and it is made up of phospholipid bilayer. Bilayer means 2 layers, so 2 layers of lipids here, and this layer is purely viscous. It does not contribute the RBC, as we said earlier, that it is a viscoelastic solid, but this lipid bilayer does not contribute towards the elastic behavior. Elastic behavior comes from membrane cytoskeleton, which is a network of proteins just below the lipid bilayer, and this is responsible for the biconcave shape. It gives the shape, whatever shape the RBC have, and it is responsible for the shape. Biconcave discoid shape, it is responsible for the shape of the RBC. And the thickness of this membrane is about 80 nanometer. Inside the red blood cells, it is filled with a liquid solution, which is saturated hemoglobin solution, which is about 32% by weight, and the viscosity of this hemoglobin is about 6 centipoise, which is about 5 times. If you compare this with the plasma viscosity, so this is about 5 times of the viscosity of the plasma. The quantity or the of hemoglobin in man is about 15 gram per 100 ml of cell, and in woman is about on average 14 gram of hemoglobin. So, as we said earlier, that the function of hemoglobin is to carry oxygen, and 1 gram of hemoglobin has the capacity to carry about 1.34 ml of oxygen. So, let us work out some numbers and find out how much oxygen can be carried by 100 ml of blood. So, if we want to calculate that, we know that 100 ml of blood has 15 gram hemoglobin per 100 ml of blood, and 1.34 ml oxygen can be carried by 1 gram of hemoglobin. So, that will turn out to be 15 into 1.34, which is about 20 ml oxygen that can be carried by 100 ml of blood. Okay. So, 100 ml of blood in a normal human being can carry about 20 milliliter of oxygen. Another property of red blood cells, which is rheologically very important, is called Fahræus. It is the property of the red blood cells to combine and make stack like that of coins, and this happens at low shear stress. So, what you see here on the left hand side is at low shear or even at rest. So, they form a network of red blood cells at low value of shears, whereas at high shears the RBCs are separated from each other, and this process and these stacks are known as rollers. The RBCs, they can take any shape under the dynamic conditions when the blood is flowing, and they can tumble, slip, can have a chaotic motion, can roll, swing, and tank trade. So one of the motions that is important is tank trading, which means that the wall of the RBC moves itself. So it rotates itself like the belt of a tank, and different points on the RBC, they take turn. This point which of the membrane which was here, after some time it comes here and then it moves to the another point. So basically the points on the membrane, they rotate themselves like belt in a trading tank. The next important constituent of blood is white blood cells. Their volume is very small compared to RBC. They constitute about 1% of the total blood volume, and because of their very small volume as compared with red blood cell, they have very little influence on the rheological properties in large arteries, where the size of the channel or size of the artery is very large compared with the size of cells. The RBCs, they are spherical in shape and their diameter is about 7 to 22 microns. There are 3 different kind of white blood cells, and they can have different sizes. In smaller channel, where the size of the channel is about the size of the capillary, or size of the channel is about the size of the cell, that means the capillary size is about 10 micron. In these capillaries, the rheological behavior is determined by each blood cell that is passing through the channel. So they may play, or they can play an important role in the flow resistance, in determining the flow resistance in small capillaries. The surface area, as you can see, is not smooth, and they are much less deformable than red blood cells. So they are almost rigid. It is not very much known about the rigidity, but what has been seen, that if a RBC and a white blood cell, they collide, the particle that deforms is red blood cell. So it is clear that their deformability is much less than that of RBC, and they have viscoelastic interior. The third important particle that is suspended in blood is platelet. Their volume is about 0.3% of the total blood volume, and they have a round or oval discoid shape, as you can see here. The size of the platelets is about 2 microns, and when they are brought into contact with ADP, they have a tendency to aggregate and form the thrombus. So they have a very important role to play in the blood clot formation. So even though in the normal flow of blood, platelets do not play an important role in determining the rheological behavior. However, in understanding the blood clot formation process for different regions, for example for hemophilia or for thrombosis, it is important to understand the flow and margination of platelets. Okay, so having looked at the mechanical properties, shape and size, numbers of or the volume fraction of different particles that are present in the blood, let us look at the rheological behavior of the blood. So in this graph, what has been plotted is the viscosity of or apparent viscosity of blood as a function of shear rate. So apparent viscosity is nothing but shear stress divided by the strain rate or shear rate, and this has been plotted with respect to rate of shear. So you might notice here that this is not constant. The viscosity of the blood is not constant with change in the rate of shear. So the viscosity is shear dependent, and you can also notice that as the shear rate increases, the viscosity decreases. So this behavior, if you remember from the previous lecture, this behavior is known as shear thinning behavior. With the increase in the viscosity, with the increase in the shear rate, the viscosity of blood is decreasing, that is shear thinning behavior. Now, if you notice at high shear rates, which is above 100 per second, the viscosity of blood is almost independent of the shear rate. So at 37 centigrade, this value is between 3 to 5 centipoise. Further, there is no effect of shear at these high values. However, at low shear rates, which is below 100 per second, the viscosity increases drastically with decrease in the rate of shear, and there is very steep increase after shear rate of 1 per second. Let us try to understand why does this happen. At high shear rate, as we know that the blood is suspended with the red blood cells, which are flexible disc shaped particles. At high shear rates, what happens? RBCs, they orient themselves at high shear rates, the RBCs, they orient themselves with respect to the flow. So the larger dimension of the RBC, that is aligned with the flow, and the resistance to the flow is minimum. So the viscosity is lower, and further increase in shear rate does not affect the alignment of the RBCs. So that is the reason that the viscosity is lower, because when they are aligned with the flow, they offers the minimum resistance to the flow, plus with the increase in shear rate, there is no further change in the alignment. However, at low shear rates, what happens? That the RBCs tend to aggregate. So at very small shear rate or at rest, RBCs, they have formed a network of red blood cells, or they have aggregated, and with the increase in rate of shear, they the network start breaking up, and at a particular value of shear stress, the blood starts flowing, and that's where that value will be known as the yield stress. And after the network is broken, the particles or the RBCs, they start to orient themselves with the flow. So that orientation is happening until about 100 per second of shear rate. So that is why the viscosity of the blood is changing or decreasing with the increase in shear rate up to 100 per second. Okay. So the aggregation of the RBC, that determines the viscosity of the low shear rate, or that is the major factor that controls the rheological behavior at low shear rates. Okay. So let us look at the effect of volume fraction of red blood cells on the viscosity. What you see here on the x-axis is hematocrit, which is volume fraction of RBC, and on the y-axis is mu blood, or the apparent viscosity of blood normalized by the viscosity of the plasma. So that is why it is called relative viscosity, that is viscosity with respect to the viscosity of the plasma. So as you can see from here, the viscosity of the blood increases with respect to the with increase in volume fraction of red blood cells or with increase in hematocrit, the viscosity of the blood increases. At one particular hematocrit value, with the increase in shear rate, the viscosity decreases. However, there is not much effect of the shear rate from 64 to 212. In the same graph, another line has been plotted, which is the viscosity of a solution which contains rigid spheres, and you can see that for the same volume fraction, the viscosity of a solution which contains rigid spheres is significantly higher, about twice than that of blood which has flexible and disc shaped particles. So, this clearly suggests that the flexibility and shape of particles that is responsible for minimizing the viscosity of the blood. Now, as I said earlier, that the hematocrit or the percentage of RBC is also responsible for the yield stress behavior of the blood, and it has been shown that it depends on, experiments have shown that it depends on the concentration of fibrinogen as well as concentration of hematocrit. If there is no fibrinogen, then actually the yield stress disappears, and the Rayleigh formation or Rolex formation is also not seen if there is no fibrinogen present in the blood. On this graph, the yield stress to the power 1 by 3 or 1/3 power of yield stress is plotted with respect to the volume fraction of red blood cells, and it is seen that the value of yield stress increases with increase in the hematocrit value or the volume fraction of RBCs. At below a certain RBC volume fraction, the yield stress is 0. So, that value can be between 5 to 8% or has been seen in experiments to be between 5 to 8%, below which there is no yield stress behavior observed of the blood. Okay. Another important effect which is seen in the rheology of blood is the effect of the size of the channel. So, as you can see from this graph, that the relative viscosity, again the viscosity of blood with respect to the viscosity of the plasma, it decreases with decrease in diameter, and this behavior is observed only below certain channel diameter. Okay. So, it is seen that there is no effect of the channel diameter for up to 0.5 mm. Channels, for the channels having diameter 500 micron or 0.5 mm or more, there is no effect of channel diameter. However, for small diameter capillaries, especially less than 300 micron or 0.3 mm, the viscosity of the blood decreases with the channel diameter, and it happens so until the diameter of the channel approaches towards the size of the capillary, which has same diameter as that of the red blood cells, about 6 to 8 microns, and this effect is known as Fahræus-Lindquist effect. Okay. Why does that happen? This is explained, or this has been explained in the literature, that when the red blood cells flow in a channel, because of the velocity profile or the velocity gradients, they spin, and then spinning RBC, they have a lift force that push them away from the wall. So, the RBCs always have a tendency to move towards the core of the channel, and in this core, so the red blood cells move in the center of the channel or core of the channel, and the near wall is the layer near the wall is only plasma. It is called cell free or plasma skimming layer. As we have seen that the viscosity of the plasma is about 1/3 of that of the blood, and the flow resistance in a channel is determined by the viscosity of the fluid which is near the wall. So we can see this in this manner, that in the core there are RBCs, and this is cell free layer, and this is core. So in small diameter channels, because the thickness of the cell free layer is almost constant, in a small diameter channel the cell free layer becomes a significant part of the core region, or a significant part of the channel cross section, and it helps in minimizing the flow resistance or the viscosity or apparent viscosity of the blood. This effect is known as Fahræus effect, the effect that in small diameter capillaries RBC concentration is decreased with decrease in diameter, and RBC there is a cell free layer near the wall. This effect is known as Fahræus effect, and this can explain, or this is one of the causes that can explain Fahræus-Lindquist effect, that is decrease of the blood viscosity in small diameter channels. So in summary, what we have studied in this lecture is that the viscosity of plasma, the components of plasma, the mechanical properties and concentration of red blood cells, which is also known as hematocrit, they are major determinant of the rheological behavior of blood. The blood behaves as a Newtonian fluid at high shear rates. At low shear rates, a shear thinning behavior is observed, and two major factors are responsible for it. One is aggregation of RBCs, the another factor is shape and flexibility of RBCs. Okay, and then the viscosity decreases with decrease in capillary diameter. So this diameter is, if the channel diameter is between about 10 to 300 microns, then the viscosity decreases with decrease in capillary diameter.